Okay, so let's start today's class. So, if you can hear me again, please raise your hands so I can check. Okay, so at least two people can hear my voice, so it's working. So we have few questions from last class about the some concept, and I thought I missed one explanation about the penetrance. So the question from Means about the penetrance, the point of the question is actually you have to understand the layers of the concept of fitness and fecundity, but the terminology itself for the penetrance is indicating the ratio of how many people having the genotype and the phenotype as well. So if you look at certain mutation, in this context, it has to be allele, right? And how many people has that condition given the alleles, right? So if you look at 100 people, and if you look at all of them has the allele for certain mutation, and 80% of them have the condition, then you can say that 80% the penetrance for the mutation, right? So penetrance is the indication of how many people has that condition given the allele, right? And the condition could be deleterious or beneficial. So it's any trait you can describe, right? So it can be disease, it can be just the normal traits without any severe conditions. So this is basically meaning the traits. So how many times that alleles appeared in the population. So you have to understand the concept of penetrance in that context. Okay, so move on to today's class. So where are we now? Give me a sec. I got a call, so sorry for that. Sorry, student. I got a call from the institute. So okay, so let's move on to today's topic. I got a message from some students about the about the class. Okay, so you guys have the what is the midterm exam period, right? So let's start from the 20th April, right? 20th April is the next next Tuesday. Okay, so midterm exams start from next Tuesday, and also it continues to 3rd May, right? I mean, you can confirm it, or if you have the alternative, the schedule, please let me know through the Zoom chat or Slack. So the point is that we check the attendance through the Zoom, and it's automatically stored in the Zoom system. And if you miss 1 or 2 class, I don't think it will affect on the grade, because the attendance is up to it's 1/3 missing in the class. So that case it will be missed out, but otherwise it will be okay. And also the all of the classes we've done so far it's on the YouTube. So right, so if you go to Slack, and if you go to one of the YouTube video link on the Slack channel on the general channel, then you can go to my account and you can follow up the latest video, right? So let me copy the link and going into here. Okay, so and press the dislike and you go to this channel, right? So then you can see, you know, the latest video lectures for our classes, right? So this is one last week, and this is one for the last Monday, right? So only one. Oh, okay, so it's almost 7 people watch the one. Okay, so I mean, if you have the midterm exam on the class day, so you can schedule your time, and also you probably have your attendance record. So I don't think you're going to be a problem with that situation, but because you're not going to miss the 1/3 of the class during the semester. So check your attendance, then if it is okay, just miss 1 or 2, then you can just follow up. Okay, so that's on your midterm exam schedule. And I mean, we are going through the class anyway during the midterm exam. I mean, of course, I mean, you're not gonna you don't want to get the class over the midterm exam period, and I'm too, right? So I'm always dreaming the missing the class as you, but you know, the university always watching me and watching us, and they are tracking our progress, right? So because you know they are managing the university system, so they are watching how lecturers or professors doing in the class. So that's why I cannot miss any class over the midterm exam. But you know, I'm on your side. So okay, so we are on this session today, and this one will be the next Monday and next Wednesday. I mean, if we are not delayed on time, and we're going to have the review session on after the next week, and the Wednesday is exam, right? So this is the session 3 schedule. Okay, so back to linkage disequilibrium and LD and haplotype, today's topic, right? And is he still on here? This guy was really fun, but I think he disappeared sometime. Okay, so what are we going to learn today? We're going to learn LD and haplotype, the basic concept, and then we're going to discuss how we discover these LD or haplotypes. Basically, the same concept and how we assess them. And also we are discuss about the project called International HAMMAP project, which is the first large-scale attempt on the identification of LD in human genome, right? And this is the lecture material, so you can look up for your learning. Okay, so LD and haplotype. So as we learned in the session 2, the alleles or genes are linked together, and they are sometimes transmit together during the meiosis, and they are segregated to the next page. And we know that this is the denial of the second laws of the Mendelian inheritance, and when we call this phenomena as the linkage, right? So, or the genetic linkage. So LD is representing the situation how this genetic linkage happening in our genome. So LD is basically meaning the non-random association of alleles at 2 or more loci in a given population. And this LD concept was first used in the 1960s by Lewinton and Kojima. Of course, we know about the genetic linkage because of the Morgan's experiments, but the formal the terminology introduced in the 1960s when population genetics discussed in our field. Okay, so LD is between 2 alleles, is related to the time of the mutation event and genetic distance, like a physical distance on the chromosome, and also the human population history, but also the other natural organisms population history. And we often call this LD as a haplotype, and haplotype representing a group of alleles in an organism that inherited together from a single parent. So haplotype is the region where linkage disequilibrium actually happening. So LD, if you think about the LD, it's always has to be understood in the concept of the coalescent theory. So how these these genetic regions are inherited from the ancestral generation to our generation, right? So if you look at the ancestral mutation on a founder's chromosome, so I mean, the founders is the sort of the first person or first people in our population, and we kind of really pointed out who they were, right? But I mean, we can assume that there must be a founder people in our population, and we call them the ancestral chromosomes. And there must be a mutation in that individual, right? And if you look at that mutation on the founder's chromosome, it has to be inherited to the present generation, which is you, right? And this mutation has the forms of LD because this mutation is sitting in somewhere in the chromosome, and that region has to be forming the genetic linkage with the other regions, right? And I mean, always the rule is the physical distance is close to adjacent to each other, then it's high chance to having the same the alleles in that region, right? And chromosome stretches are derived from the common ancestor. All of the mutation chromosome are shown in the light blue, like in here, right? So these regions are segregated together during the meiosis and next to the next generation. And also the new stretches introduced by recombination are shown in the dark blues, right? Like this one. And markers are physically close, tends to remain associated with the ancestral mutation. So as I mentioned, that if the mutation here, then probably the next generations, they are likely to have sort of the same genomic regions, which are closely located in on the chromosomes, right? So that's why there has to be a high chance of having the segregation in the linkage situation, right? So in other words, you can think about that if the mutation happened in an ancestral allele, and the mutation around the mutation has higher chance of having the genetic linkage, and that area has to be segregated to the next generation. So ultimately, these regions, like the blue light region, which is likely linkage, they are forming LD blocks, or we can say the haplotype block. And if you look at the present, the generation's genome, then there must be a lot of the block types region which are forming the LD patterns in our population. Okay, so if you want to measure this LD block and how frequently they are happening in our population, then you can use the frequency of an allele in a population, right? So you can look at the two alleles frequency, so at the two loci, right? So first allele, which is the allele A at the first locus. So you can imagine that these are located on the left side, and the frequency of the allele B at the second locus, which is the on the right side, right? So you can assume that these two loci can be linked to each other, and it can be subject to the linkage situation. And also, these two allele frequency is the independent one, independently measured to each other. And also you can measure the frequency of allele pairs. So this time you measure how much they are segregated together in a population, right? So basically you can measure how often LD happen at the region, which is the probability of the allele pair frequency, which is PAB minus the frequency of A and the frequency of B, right? So PA and PB, right? And this is D, means that the coefficient of LD. So how much LD happening at the position, right? So if DAB equal to 0, this means the equal, right? And we can say that the allele A and B are totally linkage equilibrium. So that case, these two alleles are not linked to each other because they are totally independent to each other, right? So the frequency of allele pairs are same as the frequency of A times frequency of B, right? But if the DAB is not equal to, that means not equal, then you can say that the allele A and B are somehow linked to each other. So we can say these two alleles are in the linkage disequilibrium, right? But in this measurement, you have one issue about the scales of the frequency, right? So if you think about the alleles in the population, some alleles has the higher frequency, some alleles has the lower frequency, right? So I mean, this calculation has to be linked to the has to be influenced by the frequency of allele. So that case, you have to think about the different ways to make the same comparison, make the fair comparison, right? So in this case, you can do the normalization, right? So normalization in the most of the natural science is it's a situation you put in some scale, right? Because the measurements are different by the each trial. So that case, you have to put it into the same scale, then you can compare between these two, right? So in this sense, when you compare the LD measurement using the high frequency in a population or low frequency in a population, then your the measurement of LD, which is the D coefficient, is differ by each other because of the frequency of the allele itself, right? And this is not fair comparison for the calculating coefficient. So this case, you calculate the normalized value, right? So how you do that? So when you measure that LD coefficient, then you measure the max D information from the region. Then you calculate the max D value and divide by that value for each D values in the region. So if you think about the entire the genomic region, then you have lots of the alleles within the region, and some of the alleles has the high frequency, and some of them has the low frequency. So when you measure the pairwise information from the two alleles, then you can calculate the maximal values from the coefficient. Then you divide by the value for entire coefficient value. Then you can put it into that values into the normalized scale, which is range from 0 to 1, right? And if you do the normalized LD value, that case you are not sensitive to allele frequency, as I mentioned, that the whatever the high allele frequency or low allele frequency doesn't really matter, right? And or population geneticists also developed a new method called R-square, which consider allele frequency and has ranged between the 0 and 1. So this is the based on the correlation value between these two alleles. So basically, the quite similar concept as we used in the normalized LD values. Okay, so let's look at the DNA sequence from 5 individuals. So you put it, you collected 5 people from the population, right? And each person has their own DNA sequence, and we put it into haploid information, right? So one strand of them. So this is the first person, second person, third person, fourth person, and fifth person. And if you look at certain area, if you look at this area, and these 3 people has the A allele on this locus, but these two people has the T allele in this position, right? And we can assume that these are the main alleles, right? And sample 4 and 5 also has the main allele, which is G in here, right? And you can assume that these T and G appear together. So we say these two are linked to each other, right? So that case, you can expect that these two people has the LD blocks on this pattern, right? So if you want to measure the LD, we can simply put it into the concept of the haplotype, right? So we have the different phenotype individuals. So some people has the black eyes, some people has the brown eyes, some people has the blue eyes, right? And you can put it into the haplotype information as we put it into the alleles in previously learned, right? So if you look at 2 SNPs on here, so 2 alleles, two locus here, then you can say that this person, the individual 6, has the black eyes, has the A SNP on this position, and this person has the G on this position, and black eye A, blue eye A, and brown eye G and G, right? And then this person has the G on the second SNP and T, G, G, T, T, right? And then you can expect that the A, G, and nothing on here is the black eyes, or G, T has the brown eyes, and the blue eyes has the A, G, and A. Okay, so this one has to be here. I have to mark in here, okay. So these 3 phenotype has the different haplotype composition in their genome, right? So it's again another SNPs on this position. So let me quickly fix this one. Okay. Sure. Okay. Cool. Right. Okay, so if you look at the 3 SNPs, then you was my bad. This is not SNP. It's deletion, right? Because this is the in there. Okay, my bad. Okay, so if you look at the 3 loci, then you can assume that which haplotype forming which phenotype, right? So if you look at more global scales on the chromosomes, then you can look at more SNPs onto the haplotype regions, right? So this is the scale of 150 kilobase in our chromosomes, right? And each individual has the unique haplotype composition in their genome, right? And also it's more specifically, it's forming by different ancestry. So it's different by different populations. So if you look at certain population, maybe from the Asia, or maybe from the Europe, or maybe from the Africa, then each population has their own unique compositions of the haplotype block and haplotype, the SNP compositions. And when you measure that the different combinations and calculate the haplotype frequency, then you can estimate, the individuals belong to which population, right? So that was the one features of the haplotype, and it's fully representing the ancestral information. And also, if you look at different population in our species, then African population have generally smaller blocks, so which are about the 7.3 kilobase per block, but compared to Europeans, like a 16.3 kilobase, or Chinese or Japanese, they have 13.2. The reason why they have the different block size is that the African population has much larger sample size, much larger population size than non-African population because of the, the migration history. So that's why they have much higher genetic pools, so they can shuffle up their genotype information, also have a greater chance of shuffling their genetic information in the genetic recombination situation. Also, if you look at the haplotype, then you can specifically measure how frequently they are happening in our genome. So this is the same topic we discussed about the coefficient of the LD in the slide 9. So this is the actual examples of how we represent that the LD information into our genomic position. So this is the pairwise position of the chromosome. So this is the chromosome position, like from the 1 to 14. So this is the very simplified version of the chromosome position. So we can only looking at 14 loci for the 14 SNPs, right? So just removing the distance from the chromosomes. So we only put the loci numbers, right? So this is the one way. And this is the another axis, which is the same strand as this one, just put in the pairwise comparison, right? So if you look at the relationship between 1 to 1, then it always has the 100% LD information because it's the same loci, right? And also you can compare 1 to 2 or 1 to 3, or sometimes like this black square, which is the 2 to 7, right? So position on 2 and 7, how much they are segregate together, right? So it's also 100%. So if you look at the blocks like this is the LD position from the 1 to 8 position. So from here to here, so they are high chance of having LD blocks, right? So if you measure that LD coefficients, they are always like 100%, which is the 1 in here. And if you look at the 11th to 14th, which is from here to here, is also having the LD formation too. So if you look at this chromosome pattern and put it into pairwise information like in here, then you can imagine that these two are the LD formation, right? So I think some of the students are not familiar with the pairwise formation, right? So we can put it into different concepts. So let's turn around. Okay, go to front. Oh, damn. Not this way. This way. Oh. Because we don't have whiteboard in this class, so we have to be like this way. Okay. So this is the same concept we look at. We look at the previous image, right? So this is the 1 to 8 comparison. Has to be scaled in some way, like in here. So okay. So this is not really matching, right? So 1 1 2 2 3 3 4 4, right? So this is the same intervals on this plus, so it's a little bit distorted, right? But anyway, these formations, so from 1 to 8, they are forming the LD blocks, right? This position. Oh, sorry. This position, right? And this 11th to 14th, so they are somewhat here. Okay, so this position also forming the LD blocks too. Okay, so this is the one way we can look at the how to loci looking at LD blocks in the genomic position. Okay, so this is the first evidence of the haplotype blocks in our human genome data set. So that's the study from the 2001, and the first time people looking at the haplotype patterns in our genome, and what they found the patterns of the LD in a 200 kilobase region of the class 2 region of the major hit. So complexity complex in humans. So that is, I think, is chromosome 6. And people looking at how the genetic loci they are linked to each other, and how big they are in our genome. And this is about 200 kilobase. And also they're also using the D prime information for the LD coefficient. And as I explained in a previous slide, D prime 0 is the totally linkage equilibrium situation. And if the D prime is close to 1, then that is the LD situation. Okay. Something going on outside the Hana Science building, and they cracking the building for using the water. Okay, so, they basically clean up the wall. Okay, so back to the haplotype block and TXNIP. So if you think about the haplotype blocks, it's like a representation of the genomic regions in our chromosomes and genome, right? So so I can compare this concept with, like, the regional sections in the city, like, if you look at the Seoul, we have like a Seongbuk-gu or whatever, the Gangnam-gu or whatever the locations, right? So if you look at the chromosomes, you also have sort of the haplotype regions, like, we define in the city, right? And if you look at that haplotype region, you can pick up 1 or 2 genetic positions to represent the haplotype, and we call it TXNIPs, right? So this is the representative SNP in the regions of the genome, and that SNPs has the sitting in the high LD the regions and representing the groups of the SNP within the haplotype, right? And or sometimes we called it, these TXNIPs, we sometimes called it markers or SNP markers, right? So why we need these TXNIPs? At the first time people looking at the haplotype, so let's imagine that, the 2001, at this times, looking at one person's genome is, like a cost millions dollars or $10 million, so which is amazingly expensive, right? So people developed a new technology, which is much, the cheaper one, which is the microarray. And people want to look at the few SNPs in our genome rather than entire SNPs in our genome. So if you know about the haplotypes, then you can pick up the representative SNPs in the genomic region, and you're looking at the TXNIP information, then you can infer, which population samples from, or which haplotype samples have in the regions, right? So mainly using the TXNIP was to reduce the SNP genotyping cost. So you can screening the lots of people at the time, and also using that information for the other disease studies, like an association studies. And then at the first time, people looking at quite few SNPs in the early days, like 450K, which is the 450,000 SNPs in our genome. But before the, around the 2001 or 2002, we can only look at 100 SNPs or 1,000 SNPs. And after that, lots of commercials coming up, and they develop like a SNP array, which can look at the 450 SNPs or 700K SNPs. Nowadays, people look at around 2 million SNPs in our genome. And also, we have better situation like, we can look at all possible SNPs in our genome using the genomic sequencing technology. Okay, so how we discover an assessment of haplotype using genomic information. So there are two ways to look at the haplotypes. So first one is the direct approach, and the second one is the indirect approach. So direct approach is read all nucleotides of your genome. So basically, you're looking at all SNPs in your genome, all variants in your genome, right? And indirect approach is you infer, so you basically guess which haplotypes you have in your genome using the text SNP information. So for the indirect approach, the key concept looking at that haplotypes or guessing the haplotype is the common SNP, right? So common SNP means that the commonly present in our population, and that is the types of the single nucleotide polymorphism, which we learned in the previous session. And how common defined is the frequency threshold greater than 1% or 5%, right? And this common implies that we know the frequency of the variants, so that's why we can call it polymorphism. And also there are millions of common SNPs in our body, in our genome. So many SNPs we can use for whatever disease studies or association studies. And also, as I mentioned, they're looking at all possible variants, which are whatever common or rare, will be quite expensive. So looking only a few portions of them, so you can easily utilize that information for the haplotype inference, right? And the other way is the direct approach, which is looking at all possible nucleotides of your genome. So basically, if you have the 5 papers sequence as we discussed in previous slide, then you can basically read all possible SNPs from the population. Then you can infer the haplotype information from that. And if you want to look at all the nucleotides in your genome, then you basically genotype all, the 3 billion position in our chromosome. So it takes lots of time and also cost as well. So technology we can use for this direct approach is the Sanger sequencing or next generation sequencing. And depends on the technology. We can finish in short time, or it takes like almost forever. Or indirect approach is read only small sets of the text SNPs. So text SNPs, as I explained, this is the SNPs representing LD blocks or haplotype. In this case, you use the technology called microarray, which is you put the text SNPs into the chip, like an array type platform. Then you can read out the text SNP information from lots of samples, right? And this is much efficient and comprehensive approach. So if you look at 5 people's sequence, you tag up some SNPs. So like in here, C and A. So you use these 2 position as a text SNPs, then you only read these 2 information rather than reading all of the nucleotides in that area. Okay, so first one we can discuss about the direct approach, which is the Sanger sequencing. So this is the method. This is one of the traditional method. You can look at the DNA sequence from the genome, and you use the primer and fluorescent image. Then you can bind that each nucleotides into the template. Then you can read out which nucleotides forming in each position. So you have the nucleotide information representing the T, C, A, G, and put the fluorescent with colors, right? And then you attach each of the position on here. Then you can read out, okay, this position has G, and this position has G, and this position has A, this position has C and T, whatever. And then you measure that, you know, each positions are nucleotized, and you read out the sequence information. But saying sequencing is old technology, and also you can only read some 100 base pairs. So if you want to read 3 billion base pairs in your genome, it takes almost forever. So if you start today, then you're not going to finish until you die, right? So this is one of the old technology, and we're going to discuss how we overcome that situation in later section. And another technology we use for the indirect approach is the microarray. So as I mentioned, that this is like a chip style or array types. So this is like these tiny blocks like in here. So this is about your 2 fingers, and you insert that the text needs onto the position. So if you know the sequence of the tag position, then you bind that tag SNP probe onto the solid panels, which is like this chip or array. Then you put your DNA, then which DNA parts actually binds to this area, then you can estimate which sequences is binding to information. So if you compare the Senya sequencing and microarray, what we can think about is the Senya sequencing only can read 1 or 2 positions of the genome, because it can only read, you know, 100 base pair or 1,000 base pair. So Senya is you know, well read for the entire nucleotides given the region, but it only look at small regions of them. But microarray is basically reading all of the position of the chromosomes, but you look at the positions partially, right? Because you choose 1 or 2 SNPs from the haplotype, right? And you put it into the chromosomes, then you can basically read all entire chromosomes, but looking at all, you know, only sparse loci into the chromosomal positions. Okay, so TextNIPS was initially developed by some people in 2000 and 2001, and they tried to look at the haplotype patterns using this TextNIP information. So one paper from the Mark Daly's group and Gabriel's group in 2002 and 2001, and they're looking at the TextNIP information and how much the LD forming into our genomic positions, right? So how they did at the time. So let's imagine that we're looking at this position, and we have 5 people on here, and you have 7 loci in this region, right? And we know that this position has the haplotype blocks, right? And we have the 7 loci. So I put S1, S2. This is a SNP1, SNP2, SNP3, blah, blah, blah. And the major alleles is the gray square, and the minor allele is the red squares, right? And this, okay, so this P1 is the population 1 and population 2, population 3, and population 4. So population 1 has the minor allele on the SNP1 and SNP6, and population 2 has the minor allele on 2nd, 4th, 6th, and 7th. And population 3, position 1 and 3, and population 4 has 1, 5, and 7. And they uniquely have their minor allele composition in the positions, right? And if you look at certain individuals' TextNIP information from the microarray data, and this individual has the snake composition like this. So it has the minor allele on the 1st position, and 5th position, and 7th position. So what you want to do that, you want to tell, I mean, which haplotype people has, and how you can define this person's population, right? So if you look at this SNP 2, 3, and 4, then this person has all major alleles, so you cannot distinguish which population these persons belong, right? And then if you have the information, all of the 7th position, then you can basically infer some, you know, the information, but this is too, you know, the ambiguous for the inference, because sorry, okay, So 2 and 3 and 4 are, in this case, 1, 2, and 3. And if you look at the SNP 1, 2, and 3, then this is too ambiguous because the SNP 1 has the minor allele on minor allele on the population 1 and 3 and 4, right? So this is a bit of question. But if you look at the SNP on the first position and seventh position, then you can basically identify these individuals probably belong to the population 4, right? Because this is well distinguished the population information. So if you want to set the text SNPs from the population information, then you have to choose the well-known the SNPs which are distinguished the population differences between ancestry, then you can use for the text SNPs, right? If you use all of the SNPs, then that case, this is the direct approach for defining the haplotypes, and that is the inefficient way. So you rarely do that. So if you want to design the the good microarray, then you have to pick up a few SNPs for the tagging text SNPs, right? So in that case, you have to pick up the SNPs that can distinguish the population easily, right? So there is one way. And why we care about the haplotypes for the biomedical science context. So the reason is that if you want to study disease, then if you know about the haplotypes, that is much efficient way to do the genetic studies. So if you want to compare the two groups, so one group has the disease and the other one group doesn't have disease, so we can call control groups, then one way you can do is you look at all the nucleotides from the people in each group. But again, this is too laborious and this is too expensive way. So you can basically use the haplotype information if you know the haplotype information, right? So if you know that, if you look at certain area and if you know that that area has the 3 haplotypes, then you can basically measure the frequency of the haplotypes. Then you can compare these 2 frequency between the groups. Then you can point out that, okay, so this haplotype is related to the disease, right? So let's imagine that the new mutation happened on the ancestral population. So new mutation could be de novo mutation, and this de novo mutation doesn't have the high natural selection pressure. So somehow segregate to the next generation, right? And this mutation could give a risk to certain traits or disease, or sometimes disease like an Alzheimer or Parkinson's because it has the late onset, right? And this mutation could be survive over the time and segregate to the next generation, right? And this mutation has forming the LD blocks onto like this position. And over the many generations, like a 40 generation, it has the 3 haplotype blocks on these genomic locations, right? And if you then you compare the peoples with the disease at the present generation, then you can compare the disease groups versus control groups. Then you can basically measure which area, which haplotypes has the mutation alleles in their genome. Then you can easily define that this is related locus using the haplotype information, right? So I think this is one of the key figures we can point out in these sections, because using the haplotype logs for the disease study is one of the key experimental design in the larger scale genomic studies, because I mean, essentially we're not going to look at all of the haplotype, all of the nucleotides in the patient's genome. We can basically using the haplotype information and using the text SNPs and put it into the association testing. So this is the one easiest way, and how, and also these figures is well recapitulate how mutations is occurring in the ancestral generation, and how this mutation is segregated to the next generation in a coalescent perspective. So I mean, if you understand well about this plots, I think you probably okay with any concept in this session. Okay, so one of the earliest haplotype study was done by Mark Daly in 2001. I put his photo on here because he's the living legend. He's the living Gregor Mendel in our time. He's not one of the, he's the greatest geneticist in these days. Mark Daly found the haplotype patterns in 2001, and he presented this result in, I think it's in Nature or Science. It's Nature Genetics, right? And he's the person did the first the high resolution haplotype structure in our genome, and he's basically looking at 500 kilobase regions on the chromosome 5 and 5q31 region, which is known to be a Crohn's disease region, because he worked in that area at the time. And he firstly looking at the haplotype patterns across the many peoples in the world, around 129 trios, which is the parents and offspring. And they look at the direct approach, which is that they look at the all the nucleotides from the region, which is quite expensive information, and they're looking at the how well and how frequently haplotype forming in the region, right? And they look at and then this is the results from the paper. So they look at 500 kilobase region, and they look at 11 blocks from the region, and each block has around between the 3 to 100 kilobases, so small blocks or sometimes large blocks, right? And these regions are always forming the linkage area, and they tend to segregate to the next generation. And because of this haplotype information, this is the sort of the initiation of why we study haplotypes in human genome. So I mean, we're going to learn about the Human Genome Project, which is done in the 2001. And after the Human Genome Project, people right away start the new project called International Haplotype Consortium, because people want to understand the haplotype structure in human population. So yeah, I mean, I attempt to explain the TMI. I don't know. I mean, I sometimes read some, the public post or the whatever the bestseller books in the science field in our country, and some people always do sort of the rhetoric of why human genetics is not successful at the time, because they always the mention about after Human Genome Project, people thought that human DNA is not the sole structure, so that's why people looking at the epigenetics, and that was, I mean, frequently appeared in the sort of the bestseller books in the in our society. And I mean that's totally wrong, because after the Human Genome Project, we didn't do anything about the epigenome project. I mean that is, I don't know how authors came up with that conclusion, but actually after the Human Genome Project, people start doing haplotype studies. But the problem is that lots of the authors in whatever science bestseller books, they don't understand haplotypes, because haplotypes is not the topics we normally learn in the human genetics class. But anyway, and the second TMI, so this is the first paper about the haplotypes, the paper. I mean, if you want to read this, I mean, it will be interesting, but very old style. But anyway, the corresponding author of this paper is the Eric Lander. He's the chief scientific advisor in John Biden's government in United States. So he's the one of he's again the greatest scientist in our living days. So if you're looking at his lecture video in YouTube, you will be amazed. Yeah, I mean he's amazing guy. And another TMI, Mark Daly at the time, he didn't have PhD, so he was one of the researcher in the MIT Institute, Broad Institute. And Mark Daly was, I think he's 25 or 26 at the time. He was quite young, but he's really brilliant and he did lots of things. And Ed Lander asked him, you know, why don't you do the PhD? Then if you on PhD, then you can become professor in MIT. So Mark Daly went to Netherlands and he got the PhD. I think it's the remote PhD or something, that program, and he did in the 1 or 2 years. He finished his PhD and he right away became a MIT and Harvard professor. He's an amazing guy. I mean, I met him twice, and I mean that was my best days in my life, right? And yeah, so now he's in the Finland Genome Project, and I mean lots of the great genetic project from his perspective. So I mean, if you are burning desire in human genetics, you can follow up his achievement. Okay, so what happened over 15 years after we know about the haplotype logs? We developed lots of things about human traits and human disease. So one of the application from UK was looking at the haplotype log and designed the microarray, and looking at the lots of UK people's DNA using that microarray technology, and they've done the great genomic research and public health research called UK Biobank, right? And they designed their array using the Axiom company, and they call the UK Biobank Axiom Genotype Array. And what they've done with the lots of different, you know, the trait values or the genetic values, they including all of the stuffs on here, right? And that was one of the technology they used. And after UK Biobank studies, you know, we learned about lots of things about human disease, human traits, and, you know, like big companies like Regeneron or Pfizer's, they are getting into the UK Biobank dataset to develop their idea. And also there's another microarray studies, which are Psychiatric Genomic Consortium, which is again the great genetic consortium, and they design their own things, which is infinite psych array, which is 24. And this is again using lots of different psychiatric conditions in the world. And this is one of the leading genomic consortium in the world. And how about our country? We have Korean chip and the simple name, great name, and it has the K chip. We always put K in our great things, right? Like a kimchi or everything is K. And we developed the Korean chip, and this is also lots of, you know, we're covering lots of disease and traits and looking at lots of people's health condition, and also the great research using for other many public health purpose. Okay. So let's move on to the first attempt on the haplotype information, which is the International HealthMap project. So International HealthMap project is start at the 2002, which is just after the Human Genome Project done. Then the aims was to characterize the sequence variant and the frequencies and correlate between them. And also people want to look at the distribution of the LDE across the genome, right? So at the time, we have unable to look at all the nucleotide position in our genome. So which means that we cannot do the direct approach on a human genome sequencing. So people design the microarray and they look at few positions to represent the LD blocks in our populations. So this is basically a quick overview of the genomic variation in our population. Okay, so in the National HealthMap project, we have the two major white papers, which is describing the major findings for the papers. So first one is released in 2005, and the second one is the 2007, right? And this is the including the sets of haplotype text SNPs. So they are distributing the text SNP information to public, right? So people can use that information to design their genetic studies, to design their association studies, right? And at the time, of course, we didn't know many things about the LD information in human genome. So that's why people want to look at the LD information from each population, maybe from the Europe, maybe from the Asia, maybe from the Africa, right? And human genome across all the the population information, we can divide into the 200K haplotype blocks. So if you look at all the peoples in our population on the globe, then we can estimate that probably there are about 200,000 haplotype blocks in our genome, right? And also, if you look at each chromosome position, then you can easily track down how LD forms in each population. So this is the information about the Chinese population from the Beijing and from the Thousand Genome Project, which is just after the International HealthMap project. And also we have the HealthMap project information. So we have the LD information, so we can easily look at other populations information for the LD patterns, right? Okay, so strategies of International HealthMap project was recruit individuals that can represent global diversity. So they're not only looking at the people from France or people from England. So they recruit all the individuals from the lots of populations. I mean, as possible as they can do. And then they genotyping the SNPs for all individuals, and they identify haplotype, and they determine LDs between SNPs, and they identify the text SNP for haplotypes, right? So this is basically what they want to do in their project. And at the time, which populations they sampled for their project? So they have about five, So they have about five populations. So one population representing the Africans. So they recruit the tribe Yoruba people in Nigeria, right? And then they recruit the Japanese, they recruit Han Chinese from China, and they recruit the Utah people in US. The reason is the Utah people has lots of siblings and offspring because of their religious history, and also these peoples are mainly from the Northern and Western European ancestry, right? So if you go to Utah, you will see lots of Mormons in their countries, and also they recruit the people from the Northern and Western Europe, so from the France or from the Belgium regions. These two things can be comparison because these people are from the mainland of the Europe, and these people are the immigrant from the European society. And then what they found is they look at their genome using the genotype information from the microarray information, and then they compare each populations and whether they are similar or whether they are different, right? So expectation is that the population or ancestry, they are quite different to each other, but some populations are quite similar to each other. So like, if you compare the Chinese and Japanese, they are quite similar compared to the differences between the Asian to African or Asian to European, right? So after this project, people analyze these two populations jointly. So they're not treated separately. They put it into the same population like an East Asian, and they look at the same thing. And also, they also look at the recombination hotspots because this is related to the genetic linkage and the haplotypes, because the recombination rate is not correlate with the LD patterning, because LD has to be segregated by the physically closed position. So recombination has to be rarely happen in the position. And haplotype size vary across populations due to the history of migration, as we see in the comparison between the African versus non-African, right? And also the population size. And also LD correlate to genomic and biological features. So LD, low LD, which is the LD less likely happening, is associated with the immune and neurophysiological gene. But if the LD more frequently happen, that's related to the cell cycles or DNA damage response or metabolism, right? And I'm not sure how much these findings are persistent in recent LD studies, because we have much deeper information for each population. So probably the Korean population's LD information or Europeans' LD information is slightly different because of these issues. So this is again one of the topics we can use in the studies. Okay, so what are the pros and cons of the HAMMET project? So first, the benefits. So we have the sort of the good research for the LD information, and also people now know the experimental design, how we look at multiple populations using the genomic information. And people also look at some ideas of how we implement this information into the association studies. So this is one of the advantage we learned from the HAMMET project. But also the critiques was that how we define the haplotype blocks, because haplotype blocks has to be continuous as well, right? Because I mean, there's no way we can define this person is totally Asian, this person is totally European, because the information about ancestry is also the continuous, also the dynamic information. It's all the relative information, right? So if you want to define the haplotype block by population, you have to set up some rules to look at the haplotype blocks, because if you look at the haplotype information like in here, you can set up, okay, so these two blocks are haplotype, but also you can define like this, these blocks are haplotype, right? So how we define haplotype blocks wisely in the data set. So that was the one issue. And another issue is that we used the 5 population in the study, but I mean, people weren't sure about that, whether these, fully representing the human diversity or not. But, if you look at the more latest studies, it actually includes more and more studies. And haplotype studies, they have the different phases, and after each phases, they're including more populations and better technology to see more SNPs as well. So this is the developmental stage, and also more populations included in the study. So not just the East Asian, European, or African, they including the different African people, different European area, and different, whatever, South American or Southeast Asia. And every time they did a better design and also more data set, they have a better idea of how LD forming in the population. So they have much deeper information on the allele frequency, because they enlarge their the population size, so they see the deeper allele frequency spectrum, right? So they more accurately estimate the allele frequency, and also every time they include more samples, then they can compare the allele African population versus non-African population and how they differ by different allele frequency spectrum. So for instance, like the rare variants are very specific, very specifically observed in each population, because of the rare variants are recently rising up in the neo generation, and they segregate in the in own populations, right? So it also giving you the implication of why we sampling more people in the studies, and why we care about the larger sample size in the genomic information. So there was the one indication. And after this HAMMET project, people did like 1,000 genome project, NOMAD project, or other projects, and we see much deeper and wider spectrum of the allele frequency in a genomic data set. And also because of this haplotype information, now we can track that, which ancestry you have based on your DNA. So this is my 23andMe result, and I'm bloody Korean, like 94%. And this is the estimation we get from 2017, 18 that time, and I re-log in my data set. I have 99% of the Korean haplotypes because of more people contribute on this 23andMe commercial, and they more accurately working on the ancestry estimation. But one caution for interpret this result is that ancestry is not really related to the race or ethnicity information, because this is like socially constructed, and also it's like a self-identification. So you always has to be cautious about how we deal with this, the population information linked to the, the representation of your identity in the society, right? So, and also I can look at my haplotype information about where my haplotypes is originate, right? So from the Africa, migrate to here, to I end up in here. And also lots of different haplotypes, they have their own, the tracing of the migration. So this is, again, we can use that genomic information to track down human history in terms of the DNA information. Okay, so this is all about the haplotype information, and also the how we measure the LD information from the genomic data set. And we going to continue to discuss about the how DNA information forming the population structure, and how we can think about that information from the data set. And you can probably link some idea from the mutations and also the natural, the variation in the population, and the link to the population perspective as well. So if you, I hope you read some, the references I mentioned in the slide, and also think about and digest in your side. And if you are not clear about some, concepts or some questions, are always welcome to leave yours on the Slack channels, and hopefully, more discussions going on the Slack channels, so you can, tracking your ideas onto the genomic sites. Okay, so that's up, and sorry for the interruption of the telephone call on the start of the class, and we're going to continue on the session 3 and 4 next week, and hopefully you enjoy today's topic. And if you want to follow up the the class, and the video will be appeared in the YouTube soon. So, okay, so thank you very much, and hopefully you have the wonderful rest week, and see you next Monday.